This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 559, recorded on August 2nd, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today, right here in New York City, Dickson Despommier.
Hello, Vincent.
You decided to come in.
I did. I actually missed the contact. I was going to Skype in. Who needs that?
I'm glad you did, because if you had Skyped, we would have had to double up.
Well,
because everyone's here today.
We had a full house. That's right. We have a full house.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here.
And you haven't been in TWiV for a while.
Yeah, it's been a few weeks off, so I'm going to have to reorient myself here and figure out what's going on.
Think small.
Yeah. Viruses. Yeah, I've heard of those.
That's right.
Yeah, here it's cloudy.
It's beautiful here, still clear, 84 Fahrenheit, 29 Celsius, 36% humidity. Lovely, lovely day.
It's 28 here, mostly cloudy.
A different day here. Yeah.
Also joining us from Madison, New Jersey, Brianne Barker.
I bet it's the same there.
Hi, great to be here. Actually, it's the same here as it is with Alan. Also
What?
29 and sunny here.
Get out.
From southeastern Michigan, Kathy Spindler.
Hi, everybody. 81 Fahrenheit, 27 Celsius, 39% humidity.
Boy.
And from Austin, Texas, Rich Condit.
Hi, everybody. We've got 95 on our way to 99.
Now you're talking.
That's 35 centigrade and a clear blue sky. I was out on the water this morning. It's gorgeous.
You were rowing, right?
Actually, I was paddling with my kayaking with my daughter and the two grandchildren. Kathy knows all about that.
Yeah, fun. I miss it.
Yeah, it was great fun.
When we were in Austin last summer, we saw the bats, remember?
Yep.
Dixon, were you with us?
No, I didn't go.
Oh, you didn't show up till dinner.
That's right.
Because
I was a little lagged. I had just come back from China.
You already saw the bats.
And then
right?
I came home for one day, and then I had to go down to Austin,
you didn't
Texas.
come to dinner, actually, did you?
I did, yes. I was.
Yeah, it was at Chewy's. We all went to Chewy's.
I was at dinner.
Okay. The bats were pretty cool.
Right.
Although I heard people say, that's it.
Right.
I do what, 500,000 bats is not enough?
It's
Yeah. Well, you know.
There was a show, by the way, that part of a nature show that I just saw, showing how the bats actually avoid hitting each other as they come out of that space. It's not an easy deal. Yeah, how do they?
You know, they have echolocation, right?
That's true, but they're inundated with echolocation, right?
Pretty smart.
So they use a little visual also.
Well, they don't hit each other, though. That's cool.
They do not.
They're zipping all over the place. And humans are not good because people hit each other in cars all the time.
I'll bet the bats hit each other occasionally.
You were worried about airplane accidents.
Probably got aggressive bats that fly up behind you, and zip around.
That's right.
They probably got all the same problems people do.
Well, when I fly fish, I actually fear catching bats that mistake my fly for a real insect.
Oh.
And that's not a trivial thing, because once you hook one, and that's only been, I think, twice in my whole life. Then they're
not in season.
Then what else? How do you get them off your hook?
Well,
what's a keeper size for those?
We just had a rabies case in New York State from a bat bite.
I was fishing with my son a few years ago on a dock, and he cast and he hooked a seagull.
Those
I mean, what are the chances?
Birds.
What's the chance of that? So
that's actually pretty good, because it's a pretty good chance, I would think, because they're always diving down, grabbing whatever they can, and they're always right about the right altitude.
This guy is right.
Mine, yes.
This gull was hooked in the back, so it sat down in the water. It was 50 yards away.
They're huge.
So I said, reel it in. So he starts to reel it in. Then, of course, it pulls on the gull, and it flopped its wings, and couldn't pull anymore. So finally we cut the line.
Yeah.
But that gull had 50, a lot of line on it.
Like a streamer. Yeah.
Hopefully it will not get wrapped around it. But that line doesn't degrade, right?
No, it doesn't.
Yeah,
Nope.
right.
No.
Maybe it'll work its way out. I felt badly.
Well, if it's just hooked in a feather, then it'll fall out. If
Maybe.
it's actually hooked in flesh, it'll probably work its way out of the flesh.
Or maybe another predator will grab it and get it. right?
Possible.
A bull shark. Yeah, I know. That's right.
If you like this conversation, consider supporting us.
Please
This
consider
week in
listening to a
everything but
podcast about
viruses.
birds.
Microbe.tv/contribute. More on that later. I have a meeting announcement to tell you about. This is the 10th biennial meeting of the Australasian Virology Society. And I'm glad they called it that because it could be AVS rather than ASV. This is happening December 2nd through 5th, 2019, in the Millennium Hotel, Queenstown, New Zealand.
I wish I was
That's
a virologist.
summertime, New Zealand,
It's
to be clear.
right in the middle of it, and I've been to Queenstown. It's a beautiful place.
Keynote speakers Erica Ullman Sapphire, Ben Tenhoover, many scientific and clinical topics, the usual viral structure, replication, assembly, plant viruses, phages, antivirals, etc. So this is a Australasian Society, Australia, New Zealand, I suppose many other countries in that area, right?
The Pacific area.
Maybe Singapore.
Vanuatu, or you've got Fiji.
Fiji. And so you can go to avs.org.au for more information and to register.
Right.
Now, the last TWiV we recorded at ASV 2019 in Minneapolis. And the time went by very quickly, so we didn't get through half of the questions we had ready. We didn't do any picks. So I thought we'd bring over a couple of things that were fun that we didn't get to. And one of them was a question that Kathy had asked both Sid and Pat. What is the funniest thing you remember happening at an ASV meeting, not including after hours shenanigans? And they basically didn't have anything, right?
Mm-mm.
But I'm like wanting to, of course, we shouldn't be talking, so I kept it. But I want to now tell you what I had remembered. Two things I remember, two incidents. The one is the time a fire alarm went off while Don Ganem was speaking in a morning session. And this was actually University of Minnesota in 2011. And there's a video online that Matt Evans took. And so the alarm goes off. He stopped for a while, and then it kept going. I think it went off and on and off. And then he decided to keep talking, and he gave his talk. And it turns out that someone had leaned up against it, I think. So that was really funny.
I remember.
It just goes to show a little inflammation won't stop a viral.
That was going to be my pick for the funny incident as well. And I have to say, Don Ganem, for those who are not familiar with him, is one of the most engaging speakers ever.
Mm-hmm.
And he's the only guy I know who could talk through a fire alarm and people would pay attention. It worked.
For sure. It did. And then another one was 2015 at Western University. So that's when I was ASV president. Morning session, the lights go out. And I'm like, oh no, how long is this going to be? And the local host came up to me right away. He said, we're going to fix it. We're going to fix it. The speaker was Britt Glounsinger. So she stopped. And then they put this little battery-driven light at the front of the stage so she could see herself and not trip. And she said, I will now use an interpretive dance to show my results. I thought that was just great. And she started.
Great.
Isn't that great?
It's great.
Yeah.
Absolutely great.
And that power outage was over a large part of the city. It wasn't just the auditorium or the university. It was, yeah.
So how did the city somehow fixed it right away? Because it wasn't out that long.
No, it wasn't.
I was worried that the whole morning session would be shot.
Yeah. Yeah. Well, and it was interesting because Britt had done her postdoc with Don Ganem. So either then or later, there was something about it being a curse, from Ganem down to Britt. Yeah.
Those are the two funny things I remember. Does anyone else remember any funny things that
No, like I said, you picked mine.
You picked yours? Okay.
The thing that I remember about my first meeting the ASV meeting at Wisconsin was that there was dead silence during our podcast.
Yeah.
Not a burp, not a cough, nothing. They were hanging on virtually every word. And
it's usually pretty quiet except for the cans opening.
Yeah, but
No potato chips.
No potato chips or carrots or, and I was just sitting there. I was absolutely stunned, and the room was packed to overflow.
Yeah,
I was
our room
very
was packed
impressed.
too.
And I remember the first time we did a podcast together, and it was it was not an ASV meeting, but it was
ASM,
ASM
in Philadelphia.
Yeah.
3 people.
Yeah, Alan came to that one.
Yep.
Yeah, no.
3 people. That was it.
Yeah, live TWiVs have gotten a lot livelier.
I believe.
Well, it's ASV's our home, right?
Yeah.
Yeah.
Mm-hmm.
Virology. The other thing that we had planned to talk about was some reminiscence of our first ASV. So, Kathy, you want to?
Sure. My first one was at Madison, but not at the Monona Terrace. At the Union, and the reception was at this place with a bunch of glass overlooking the lake. And I was at the opening reception. And though I knew some adenovirologists that would be at the meeting, I didn't really know anybody at the reception that I could talk to. So I was just kind of standing there by myself. And behind me, I heard somebody say, "I want to introduce you to the best baculovirologist in the world, Lois Miller." And I didn't know who Lois Miller was, but I wanted to see who the best of anything in the world was. So I turned around and got a good look at her. And then it turned out that several years later, she and I both joined the faculty at the University of Georgia within months of each other. So that was kind of fun.
Mm-hmm.
That's cool. Rich, what about you?
So I was at the first meeting that took place in Cornell. And it was a great meeting for me because a pox virologist named John Holosack had been invited to convene the pox session, but for some reason he decided it was time to pass the torch, and he nominated me to do it. So I convened that pox session, which was the first such experience for me. I was absolutely thrilled and very grateful to John Holosack. I kind of blew it, and this gives me an opportunity to apologize. How many years later?
To anybody who's
35.
Who's still with us?
So, I didn't have a whole lot of experience with meetings, and one of my models was the Gordon conferences. And at the Gordon conferences, they're very casual. There's a list of speakers, and the session convener will kind of look at the list of speakers and create his own order, his or her own order of speakers, kind of on the spot, depending on what they think is the logical flow. And of course, I had an agenda of speakers at this session, and it didn't make any sense to me at all because I saw an overall flow of stuff. So I rearranged them, which in terms of the session, I think was successful, but I didn't anticipate that there would be people going in and out and going to different talks. So people came into the session expecting a certain talk, and it wasn't there. And shortly after that, there was a pronouncement made from ASV on high saying, "Don't do that." Right. At any rate, it was there were some real breakthroughs at that session, and it was just a terrific session and a great meeting. It was a great introduction to ASV. It was good.
And years later, they let you be the program chairman and do
Let is a
Put it in your
funny,
own order then.
Yeah, that's true.
So to this day, if someone doesn't show up for a talk, they wait
Yeah,
for this reason,
yeah,
so
because
that you don't
people
get
do
out of
jump
thing.
in and out. Yeah.
For sure.
This
Big
is
time.
true.
Brianne, what about you?
So my first ASV meeting was in 2017 in Madison. I'd heard about ASV for a long time, but most of the labs I'd worked in had really a bigger focus on going to more immunology or HIV specific conferences. And ASV always sounded interesting. And when I would try to look at joining or look at attending, I would get intimidated by seeing that I needed a member to nominate me to join. And so finally, when I had my own lab and was independent, I decided I was really going to make sure that I could make this work. And I finally found someone in my network who was an ASV member and had them nominate me so that I could join and attend the meeting. And I was just so impressed with the science, the way that people were really talking about kind of unpublished data and things in progress, and the community feel that I sort of wished I had made it to attend much earlier. And I've attended every year since then and have no plan to stop.
And right after that, ASV Council abolished the rule about needing a nomination letter. So for anybody who wants to come to ASV, all you have to do basically is put in a biosketch. And if you're a trainee, just some evidence signed by somebody saying that you're a trainee. So it's much easier now.
It's good. Alan, how about you?
That is good.
Mine was 1996 in London, Ontario. And that one, I think it was the third major virology meeting that I'd been to. And I was trotting my little poster of my thesis research around to conferences. But the session I really remember was the one about polio eradication. And Olin Q from the CDC was talking about the WHO CDC plan to eradicate polio by the year 2000. Pause for the laugh track. And it really struck me because as I was listening to him talk, I thought, I'm just a graduate student, but this doesn't sound workable the way they're going to do this. And then immediately when he opened the floor to questions, the picornavirologists in the room were just on him about how exactly what I was thinking. This is not going to work relying on oral polio vaccine to do this. And I think he was taken aback by the response because he'd kind of expected everybody to be gung-ho about this program that he put together. And that was what inspired the policy forum that Vincent and I wrote for Science in 1997, where we said, using the live vaccine for this eradication effort is not going to work the way you think, and which turned out to be correct, I'm sad to say.
Mm-hmm. I'd forgotten that you went to an ASV back then. When you
that was an ASV.
Oh, how about that? Well, my first ASV was 1990. That was University of Utah, Salt Lake City. Roland Ruckert had invited me. And I knew about ASV because, as I said, during the TWiV at ASV recently, at a Gordon conference in 81, Harry Ginsberg got up at a break and said, we're going to have a meeting this afternoon about a new society for virology. And I said, I don't want to go to that. And then over the years, I heard it was founded. And then I heard you had to be nominated. So I said, the hell with that. So I never bothered. And then Ruckert invited me in 1990. That's when I was here at Columbia. And I said, well, I'm not a member. He said, that's okay. We'll get you nominated because you have to go. And so that was my first meeting. And that was pretty cool. But as I said, I was a postdoc and moving here. I didn't pay much attention to societies. Although I did go to ASM as a grad student. Why? Because my PI said I should.
Right.
So I went. Right. It's usually what you do. All right. So that's the end of the ASV TWiV.
Yes.
No, because we've got picks.
We have picks.
Right.
Some picks. Yeah. I moved some of the picks over because we never got the picks last time.
Yeah, I'm looking forward to doing that.
Dickson, those are called picks of the week.
I had a pick of the week.
I know you did, but we had some at the ASV TWiV we didn't have time to get to.
Oh, okay.
Because we spent a lot of time on more important things, perhaps.
Yeah,
perhaps. We have a snippet for you today. I thought we would talk about the Ebola disease outbreak in DRC, which has been going on since August of 2018.
Yeah. And it is now a public health emergency of international concern, whatever the terminology is. It's they proclaimed it an official big deal.
Right. Which helps them get more aid and so forth.
It helps get more aid. I mean, there was controversy about it, and the reason they dallied on that was not because they didn't think it was an emergency, but because they didn't want to trigger the kind of panic and idiotic responses that we saw the last time there was a major Ebola epidemic going on, where people were talking about travel bans and this and that. And fortunately, that doesn't seem to have happened this time around because I don't know, maybe people are just getting numb to this virus.
I think that's true to a certain extent, actually. Helen Branswell had an article about that last week saying, I'm afraid people are not paying much attention, and that's going to be a problem. Yeah.
Mm-hmm. It is a article on page A8 of today's New York Times, so it is getting more recognition, at least in that press.
Yeah, there's two things that have happened recently, I think, that are giving it a little more press. One is that it's officially a year old, and the other is that some cases have turned up in Goma, which is a major population center, and that could be the beginning of a serious problem.
Yep.
Not that this isn't serious already.
I saw today that the third case in Goma had just passed away, and they have identified a fourth case.
Mm-hmm.
Right, and those represent transmission, so that's a big deal too. That was in a SIDRAP article. But Goma borders on Rwanda, and so they had temporarily closed the border with Rwanda, but then they changed that and opened it back up.
Yeah, that case was a man a minor who had traveled to a different province. He died Wednesday after having spent several days at home with his large family. And the other people was his 1-year-old daughter and his wife, who also died. Yeah, so that's transmission within the family, which is very typical. So far we have, as of 21st July, 2,592 cases and 1,743 deaths for a case fatality ratio of 67%. And there are a number of interesting resources now. One, of course, is the SITREP site at WHO.
Right.
We'll put a link to that, but they issue a regular SITREP, which is a PDF document. So the most recent one is July 23rd. Then before that, 16th, I guess it's every week, right? More or less. They're putting one out.
Those are very good.
And
those are
really
very good.
They are.
Really nice. Yeah. And they have a map, right, where you can see where in the country this outbreak is. They have an epi curve. And they break down all the cases, local transmissions, and they have nice maps and so forth. So that's a really good resource that we've depended on for a long time.
One of the numbers,
and this is just to put it on the map. This is in the northeastern part of Democratic Republic of Congo, right along the border with Uganda, I think,
and Rwanda,
and Rwanda. Right. So and this area has been just torn apart by conflict, and they've got armed militias running around and people shooting and butchering each other, and just it's chaos. On the ground,
conflict has been going on. Conflict has been going on for years, and one of the resources that we linked here that I read said that there were as many as 100 different armed groups. Yeah.
Yep. That's the Médecins Sans Frontières article, which is really good.
Very good.
And they say it's the efforts to stop the spread are challenged by the realities of fighting an epidemic in a conflict zone.
They've had several healthcare centers that have been attacked.
Yes.
Yeah.
That they've had to abandon
and burned down in some cases, not just like robbed or anything. Yeah.
And one of the numbers here that in this whole incident that really strikes me is the cumulative number of health workers affected is 140.
Yeah.
5% of total cases. I just have such amazing respect for the people who do this. To expose yourself to that kind of danger, to help your fellow man, I just think is really heroic. It's amazing.
So let's talk a little about the vaccine, because I was wondering, can the health workers not get the vaccine?
Yeah,
they are the first line for vaccine. Now, whether then if you look, so I had a little thing here, 107 as of 21st of July, 171,052 people at risk have received the vaccine.
Do we have updated numbers on any kind of efficacy measures that they've been able to do?
No, there's none.
No.
If there's anything they aren't telling us, probably I don't mean that in a bad way, but they don't have. My assumption is that they don't have numbers that they think are meaningful in any way. Right. I do know that healthcare workers are on the front line for vaccines, so it would be interesting to know whether these healthcare workers that were affected had been vaccinated.
Yeah, of course, trying to run a vaccine trial in that environment with 100 armed groups running around is impossible. Those are all the same complexities you've got with trying to stop an outbreak, and so it's totally understandable that they don't have those data. I was just asking if anything come out.
Right. I don't know, but I assume that this trial is going to be run sort of the same way as previously. I know they're doing ring vaccination and several modified ring vaccine protocols where they identify people at risk and vaccinate everybody around them and their families and stuff. And I assume that this is not any sort of double-blind random trial, but rather that there are people in those areas who would be eligible who, for one reason or another, refuse or don't get vaccinated. And in the end, they collect data on them. And in the end, they form part of a control group.
A number of people have said in the press that they believe it's made an impact, but I don't see any numbers that would
no numbers,
that, right?
No numbers,
right?
You know, and there are 3 ring strategies. They're using the classic ring, you know, contacts of confirmed cases and contacts of contacts. Then there's the enlarged ring, vaccinate all inhabitants of houses within 5 meters of the confirmed case. And then the geographical ring, they can vaccinate an entire village or neighborhood. They said they're doing mixtures of those, right?
This is not to be confused with the One Ring, which confers invisibility and unlimited power.
One ring that protects
rules
them
them all.
Right.
So, and the vaccine is simply the one kind,
Yes.
VSV based.
Right. With the glycoprotein of Ebola, and it's the one that was used in West Africa in the last outbreak, in which we talked about that the outbreak kind of fizzled out just before they got numbers that would be sufficient to really be, you know, convincing. Although,
which is good in the sense that the outbreak fizzled out, but yeah.
So I talked with Marie-Paul Keeney in Rotterdam. That TWiV went up a couple of weeks ago, and she was she ran that Ebola trial in Western Africa. She's the last author on the Lancet paper, and she talks a lot about it and the difficulties of doing that trial in an outbreak situation. Really interesting stuff. She said in that TWiV the vaccine would be licensed any day now. Okay, so I talked to her in May. I haven't seen that yet. And then she said in DRC, it's not clear why it's not completely working. So she felt it wasn't. I mean, there's still cases happening. I don't know if they're clearly not vaccinating everyone, but we don't have any numbers, right? So
yeah,
I think it's really hard to make any conclusions.
It's difficult to get a lot of information from the detailed information from what's going on. Now,
this vaccine is being manufactured by Merck, and apparently they have about 500,000 doses, and you know they've gotten almost 200,000 doses out there, and there's some. I've seen some articles saying they could run out if things get worse in the country. They could run out within the next year, and they're worried because Merck apparently moved their production facility from Pennsylvania to Germany. So they ceased operations in Pennsylvania. They moved to Germany and hasn't been yet licensed for being open in Germany.
Oh.
So
they moved just production for just this vaccine.
Yes, and good planning, right, in the middle of a outbreak.
Right.
Well,
but also there's another company, GSK, with a vaccine that hasn't been tested to the extent of this one. And there's been a lot of controversy in DRC about whether they could try it in this outbreak. And, you know, the Minister of Health, who quit a few days ago, he didn't want to test it. And that was part of the reason why he quit, I think.
Yeah, so I've read a little bit about this. He was a holdover from the previous administration, which is not a trivial issue in DRC, if you know what I mean. And there were a lot of issues with him, and then I think the president of the country was taking more direct control of the management of the outbreak, and the health minister felt that he was being pushed out, which was probably correct. And so he quit. So I'm not sure it was specifically about the vaccine, but I'm sure that was maybe one of the issues.
So now they have 600,000 doses available, but they're still worried. So they're considering a strategy of giving less to make them go longer and so forth. Apparently this GSK vaccine, you need 2 doses. And that's tough because I think it's really hard to get people to come back. Yeah, come back next week for your second dose, and boom, boom.
And how would you record that?
Yeah, I think it's really hard.
I mean, that's hard to do in developed countries.
Yeah.
Yes.
Yeah, I think about collecting data on the efficacy of the vaccine. Just collecting the data is going to be difficult.
Mm-hmm.
In late May, the amount of vaccine given to each person was halved already. And that decision was made with the approval of the government and the FDA, who said they were using too much.
Okay. But this all comes back to the reason the outbreak is persisting and the problems with getting data and the problems with getting vaccine. It's all the same fundamental problem that this is a virus that can only survive in humans in an environment of chaos.
Yeah.
Yep.
That you can't sustain an Ebola outbreak if you have a reasonable public health response that knows how to deal with Ebola virus, and you have some kind of logistics supply chain, and things are so bad in the areas where we get these outbreaks that it can persist.
But I think we could also use a couple of vaccines just to ensure the supply. We could use antivirals to treat people.
Since we persistently can't fix the underlying issues, it seems, we do need some kind of a stopgap, like good vaccines.
There are a couple of antivirals that are being used.
Yes,
right.
Experimentally.
That's what I've been trying to say.
Sorry.
So go for it, Kathy.
The MSF article mentions that there are 4 experimental therapeutics that are being used. Remdesivir, which is a nucleoside analog, and then the other 3 are either combinations or single monoclonal antibodies. So one is ZMapp that we've talked about before, and then one called REGN 3470, 7179, and a monoclonal antibody 114. They're only given with informed consent of the patient or a family member, they can be used under compassionate care protocols or a randomized controlled trial.
They're going to have to work on those names. Ask your doctor about REGN 3470, 3471, 3479.
But I understand that a lot of people are suspicious of vaccines and antivirals in this country as well, right? And so that's part of why they're not being used.
Suspicious of anybody in authority, rightly so.
Yeah.
Right.
Because if you have all these therapeutics, then why are people dying unless you can't get them to them, right?
And the therapeutic, I mean, you also need to provide supportive care for these patients, I assume. So,
they don't have a huge capacity. They don't, I think they have like 14 treatment centers, and they're not huge, so they could easily be.
And, they keep getting burned down. So,
yeah.
Yeah.
But also remember, this is much smaller than the West African outbreak so far. Hopefully, we'll not get to that state, right? That was over 26,000 cases. So this is.
It is, but it's also much larger than the sort of third largest outbreak, which is maybe 400 or so.
Mm-hmm. Yeah, it's the second biggest one, yeah. But I hope we can contain it before then. And by we, I don't mean us. Just
not personally.
The big we. The big we. And the healthcare workers should be all vaccinated for sure.
Yeah.
Right, Dickson?
Correct.
Now, Dickson, if you got a phone call and they said they needed your help there, would you go?
Sure.
Excellent. Good response.
Yep.
Anything else?
If they called me and said that, I would say,
what can I do? Why do you want me?
I also like to add a little qualifier on that one.
What's
why?
that?
Why did you call me?
Well, they said, we heard you on TWiV, this virus podcast, and we thought you'd be able to help.
How?
Talk to the patients about viruses.
Talk to the patients.
I'm making this up as I go along.
I think you would be making this up. I
just want to know if you would go. You're making qualified questions. Just yes or no. You said yes. So
I would drag Daniel Griffin along with me.
Well, if they had a good reason for us to go,
yeah,
I would go too. But I would get vaccinated.
Absolutely.
Twice.
Well, it's hard to get vaccinated against high-speed lead.
Yeah, that's a problem.
That's the complication.
That is our snippet for today. And I don't have an ad, so we have to go right on to our paper.
Right.
And I'm really glad we did that snippet because it's good. It's important, and it's good to update that.
Absolutely.
Agreed.
Our paper is from the Journal of Cell Science. And I got an email from the senior author, Roberto Cattaneo, who was at ASV. And he said, I'm sorry I missed you. I couldn't tell you about this cool paper we just published. So he told me, he said in his email, he thinks this is the most important thing he's ever published.
Well,
that's interesting.
And he's published a lot.
Yeah.
I was going to say.
I mean, his lab is responsible for the whole story about how measles enters and exits, right?
Mm-hmm.
Right.
Which is, just, to me, still astonishing.
Yeah, his lab found Nectin-4, the basolateral receptor on the respiratory epithelium that helps the virus get out. There's another receptor on immune cells that helps the virus get in. And these will be part of the story today. This paper is called Transendocytosis Elicited by Nectins Transfers Cytoplasmic Cargo, Including Infectious Material Between Cells. So this is just
Let's translate that.
Yeah.
Yeah. Why is this paper important?
It's cells transferring stuff amongst them.
Yeah.
They're
So cells hand stuff from one to the next, just directly, membrane through membrane. And it turns out that viral infectious material, measles, can go between this same pathway.
I look at it as the cells are getting close, and then when they touch, one rips a piece of the other off, and with it not only comes membrane, but cytoplasmic contents.
So it obviates the need for a receptor on multiple cell types, because even one cell type can give it to everybody else just this way.
You could, in
They
theory.
have, if the cells juxtapose and have this kind of a connection.
And
yes.
in fact, this paper delves into that very nicely.
Yes.
Now, this, as I understand, was an accidental finding.
I'm sure.
And
as all the great findings are.
It's sort of, there's one part of the paper where it kind of sounds like that.
Because, they set it up by saying, cells are known to transfer contents. But we'll see. But the authors are, the first one is just wonderful. Because if you ask the first author for something, he's going to give it to you because his name is Alex Generous. And the last author is Roberto Catania. We have actually a couple of people from Columbia, Barry Honig and Larry Shapiro. You know Barry Honig.
Yes.
And then Matthew Taylor, who was at ASV. He is a former Lynn Enquist person, and he's at Montana right now. So we have people from the Mayo Clinic, Columbia, Northwestern University, Montana State, Pasteur Institute here. And it's a preprint still online at the journal. It's not nicely formatted, which makes it harder for us to read.
But it's accepted.
It's accepted.
It has been peer reviewed.
It's been peer reviewed and accepted, and
it's very beautifully written. I love the way they explain the panels in the figures.
Yep.
Oh,
And the figures are awesome. It's really great.
They're
It's
almost
very pretty.
self-explanatory.
Yeah.
Yeah.
That's right. It's worth about 1,000 words per panel.
So transfer of cytoplasmic contents between cells is known previously.
Sure.
And this example is great. The bride of sevenless protein is internalized by the sevenless receptor in Drosophila. Isn't that great? The bride of sevenless?
Bride of sevenless, yes.
Drosophila have the best names for genes.
These are culturally embedded in television history and in the popular press.
Is it?
Because they lose their meaning after a while, because they say, what the hell's sevenless? What is that all about?
That's
Like
another one. that I
Hedgehog.
like. Hedgehog.
Yeah, I mean,
And Sonic Hedgehog.
they're
Sonic Hedgehog.
They're having fun with us.
And Fushitarazu.
I had that for dinner last night.
I didn't say sushi.
Actually, the one I like the best, the ones I like the best are the group of Drosophila mutants that have to do with memory. And so they have flies that can't remember stuff, and they named them all after vegetables, like rutabaga, right?
That's good.
Right.
That's good.
Right.
So there are examples of cytosolic transfer. And then they say, we discovered this, which seems to be mediated by these proteins called nectins. Nectins, Nectin. So there are proteins on cells. These are cell membrane proteins, plasma membrane proteins. There's nectins and nectin-like proteins. The first nectin-like protein discovered was the poliovirus receptor. And they didn't call it nectin back then.
No.
They didn't.
That
How about
person
you
didn't.
I didn't. We didn't. I have to say, look, many years later, someone renamed our protein.
They did.
And this offended me because they didn't even ask me if I thought it was a good idea. No, we called it the polio receptor because that's what we knew. And of course, it has a cell function, and people discovered other things. And in fact, Pat Spear was involved in this because she found that members of this protein family, which included the polio receptor, were also receptors for herpes viruses. And so she and a Japanese group got together and decided to rename all the proteins nectins and nectins like proteins. And I said, okay, fine, but why didn't you say, what do you think of this? Because since we discovered the first member, don't you think? But that's not how it works, right? The heck with it.
That is not how it works.
So we have this family of plasma membrane proteins. They're immunoglobulin-like proteins. They have domains that resemble the protein fold of an immunoglobulin, right? And they're on many tissues. They're involved in cell adhesion. The poliovirus receptor is involved in cell adhesion. And the nectins, there's nectin 1 through 4. And many of these nectins are viral receptors. We have polio receptor. We have the measles receptor, nectin 4, for example. And so that's what he works on, and that's what he was studying. He was looking at interactions among the nectins and discovered this new process. So
just to fill in a little bit for the less informed, so these are transmembrane proteins that sit on the plasma membrane of the cell. And so they have a big wad of the protein that is hanging on the outside of the cell and can grab onto things. In particular, in this case, can grab onto other nectins. And then they have a portion of the protein that the transmembrane portion that goes through the membrane, and then a smaller bit that's on the inside that gets called the cytoplasmic tail. And typically with proteins like this, the cytoplasmic tail can be involved in signal transferring. So if you tweak the outside, the cytoplasmic tail senses that and tells other proteins that stuff is going on, or it can be involved in trafficking of the protein if it's internalized to go to different places in the cell.
Now, so nectin 4 sits on the basolateral side of respiratory epithelial cells. And measles virus binds to it, enters those cells, replicates, and then comes out the apical side, and that's how it's spread. You sneeze it out or cough it out, and it spreads.
So the basolateral
side. Yeah, go ahead, Rich.
Basolateral side is snuggled up against capillaries.
Right.
Okay.
The bottom.
So stuff it's the bottom.
That's on the body side. Yeah.
Now,
on the body side.
When the virus infects you, it does not enter the top of the respiratory cells, but rather it infects immune cells in your lung that are moving around, and through a different receptor called SLAM.
Right.
CD150 SLAM. And
this is all Catania's work as well, right?
Catania, other people.
And other people.
There are other people involved.
Because this story blows my mind. Everybody assumed that it would a virus would just infect the respiratory epithelium, but
Now, the cool thing, here's a little side. The measles vaccine, the attenuated infectious measles vaccine, it was passaged in some other cells and hosts. It binds to a different receptor entirely. Right? And that's part of why it's a good tumor treatment for tumors, because it binds a protein that's common on tumors. So
finish the measles cycle. It gets into the immune cells.
Gets into the immune cells. They enter your bloodstream. The virus replicates in them. It spreads to other lymphocytes. You set up a viremia. You get a rash. Then the virus is back in your respiratory tract via the capillaries. It enters the basolateral side, the bottom of these respiratory epithelial cells. It replicates, and then it goes out.
Right.
Now
the receptor it uses there, is what?
Nectin 4.
Nectin 4.
So if you do a survey of all the cell types in the body, here's a question from the blue, as Rich would say, from the uninformed public. How many cell types have 1, 2, 3, or 4 on them?
I don't know. I didn't look.
How are the nectins distributed?
You can look it up.
That's exactly what I was asking.
I don't know.
And just also to finish the cycle just a little bit more, when the virus then exits from the lung, then it can be coughed out. And there's that whole thing about how long the measles virus can be out there in the air in a room where, somebody coughed it out, and it could still be infectious later. So that's part of the transmission aspect.
What I find so cool about this is if the virus just infected the lung epithelial cells, it might not get very far. because it might just infect one or a few lung epithelial cells and be a localized infection, and that's not a great way to get stuff aerosolized in large quantities. But the way it has evolved, it can't infect the lung epithelium initially. It can only infect it from the back side.
Mm-hmm. Mm-hmm.
So it has to cause a viremia to get to the back side of the lung to get out of the lung, at which point there's a bazillion copies of it in the bloodstream. So there's a lot of it coming out of the lung, which boosts the next infection cycle.
So it only does that in immune cells, you say. So is there an immune suppression associated with the early phase of measles?
Absolutely.
Mm-hmm.
Yeah.
So you could be susceptible to other infections.
You are.
Oh, yeah.
Mm-hmm.
Yes, you
So
are.
that's the really best reason for vaccinating.
That's
against
one.
measles.
There's another
that's
one.
Is to
There's
prevent
another one
measles
which
and the spread of
it's
measles.
going to come up in this paper.
Another one is that 1 in 1,000 kids with measles
They
get
die.
encephalitis
Yeah.
because the virus somehow gets in the brain. And that's relevant here because what the receptor is on neurons is not known. And so what we're going to find out in this paper may be relevant to that.
Yes.
So yes, there's immunosuppression in encephalitis. So this is a serious infection. You need to be vaccinated.
Period.
So, Dickson.
And in Germany, that's what's going to happen.
What?
Because it's a law now that everybody has to be vaccinated against measles or they can't go to school.
Hey,
It just
we
happened.
have that law
It just
here, except
happened.
we also have exemptions. Don't
Yeah, we have homeschooling.
So do they have no exemptions permitted in Germany?
Apparently.
No religious or medical. You have to have medical exemptions because there are bona fide medical exemptions.
But
right?
this is a very strict, stringent rule that was just passed.
Really?
Yeah. Look it up.
I want to because I'm afraid you're going to get email.
It was on Yahoo News. So it must be true.
Dixon, according to Wikipedia, Nectins show up on epithelia
Yes.
and in the chemical synapse of neuronal tissue.
That's it.
Okay. Well,
So that's pretty restricted, Rich.
Such as there may be more, but that's at least that.
I'm looking at some transcription data, and it looked like Nectin 1 and Nectin 4 are pretty broadly expressed, and 2 and 3 are much more tightly expressed.
So you should revise Wikipedia then.
A CNN article from July 17th says that the German parliament has not approved the bill, but the government has backed the bill.
Right. It will probably be approved.
All right. So these nectins, 1 through 4, they interact with each other on different cells. So if you have one cell with a nectin and another, the nectins will bind. The proteins will bind to each other, and the cells will therefore interact. And there are very specific patterns of interaction, like 1 interacts with 3 and 4, doesn't interact with 2. And these are part of the junctions that hold cells together. And so they have been studying these interactions by making fluorescent versions of the nectins with different colors.
Mm-hmm. One's cherry.
Cherry is one of them. And dendra is one that you can make green or red depending on what you shine on it.
Right.
And there are others as well. So you can tell which is which by the color. And that's why the microscopy is so beautiful and so useful, right?
That's right.
You can tell exactly. In the old days, we couldn't do this as well. It's really amazing. And this is how they stumbled on this, I think. So if you have cells with N1 and N3, they co-localize. So if you have a red and a green one, then co-localization would be yellow, right? Your
So
color.
at the boundary where they're snuggled up to each other, if they're binding to each other, you get a yellow boundary.
Art and science.
Art and science.
Next.
Yeah. So they see these, and they can do these with all the different nectins and see if they co-localize or not.
Mm-hmm.
And then they say cellular localization of N14 is most interesting. That's Catania there, because he talks like that. Is most interesting. So they not only saw this adhesion, but they saw signals inside of the cell.
Mm-hmm.
And they saw the N1, the nectin 1 inside of the cell.
And it was inside of the other
N4.
cell.
It was inside the N4 producing. So they have cells making these proteins singly. Of course, you have to have that, otherwise you couldn't interpret it.
Sure.
So if you put an N4 and an N1 cell together, not only do N1 and 4 associate at the plasma membrane, but a bit of the N1 goes into the N4 cell.
Mm-hmm.
And that's the beginning of this story. So I guess they saw that.
And it's strictly one direction.
One direction.
Strictly unidirectional.
Yes, with these proteins, it goes in one direction. Yeah,
The pictures
cool.
are just stunning.
And the
Amazing.
movie is cool.
Yeah.
Yeah.
The movie
So
is really
I have a
cool.
question. I knew Caesar Milstein,
Yeah.
who,
Do you want to tell us who he is?
yeah, he was the one who popularized hybridomas. And these are cell fusion products that you make, usually with polyethylene glycol or something.
And
like
why
that.
do you do that, Dickson?
Because you want to take an immune cell and fuse it with another cell type to make a combination immune, non-immune cell.
And why do you want to do that?
Make zillions amounts of your antibody.
And what are they called?
Because your immune cell is not immortal in culture, and the cell you're fusing it with is.
Thank you, Alan.
You're
So then you
making-
can grow up huge quantities of this immune cell that makes an antibody, for example,
Right.
and you can produce large quantities of antibody
Of a
in
monoclonal
cell
type.
culture.
That's
And he won a
monoclonal
Nobel Prize for this.
He did.
He sort of
Right.
did.
He did. He did in 1984.
You never guess where I met him, though. I met him in Nairobi.
Oh,
Monoclonal
It was
antibodies.
interesting, yeah.
Now, why are you mentioning this?
Well, because I'm wondering why these cells don't do that.
Don't do what?
Why
Oh,
don't they
don't
make a
hybridize?
hybridoma?
Well, he added.
No. Why don't they make a hybridoma?
because they're
Why don't
they're
they
only
actually
transferring.
fuse?
Yeah, they're not fusing.
No, I know what
They're
they're
not-
doing. They're sharing.
They're
part-
grabbing bits.
Yeah, they're grabbing. They're
So
sending.
why
those
stop with the bits because of the?
Because otherwise, you just be one big syncytium.
What do you want? Yeah, the fusion
Yeah.
is. You got to watch out for fusion. You have to control it.
No, you're not giving the right answer.
If you don't
because-
control fusion, you get Chernobyl.
No, it's not.
Nectin Chernobyl.
What biological feature of the Nectin fusion product doesn't mimic a hybridoma fusion?
Well, there's no. These are not fusion proteins per se.
Okay.
So there are some proteins that cause fusion, like
So-
viral glycoproteins will induce fusion.
do the light chains?
Remember the proteins involved in gamete fusion that we talked about also? They have specific properties that
Yes.
engender fusion, but these don't. These are not fusion proteins.
This is
Is it-
leeching
is it-
off
Is your idea
pieces of
here,
the cell.
Dickson, that the lipid bilayer is not fusing?
Right.
He wants to know why
It's
isn't
just
fusing.
nectins fusing.
The nectins are grabbing onto
grabbing
each other, and they
each other.
initiate a process
they're-
where when you
Right.
have this unidirectional transfer,
I got it.
you actually get, in essence, a vesicle
right.
that is budded from one cell into the other
Yeah.
Got it.
and then sealed off. So you wind up with a double membrane vesicle in the recipient cell.
It's called transendocytosis.
There you go. Transendocytosis.
It appears in the title of the paper.
In
So
fact-
So it's not really, in that respect, it's not really a fusion event where you're getting fusion of the
No.
membrane.
okay, so it's a
Right.
form of phagocytosis.
It's nibbling.
Yeah.
It's
Nibbling.
almost
There you go.
phagocytosis,
They're just swapping spit.
but only in one direction, Alan.
Yes. Well, okay. Yes.
And in fact, they ask, is this endocytosis? And they use proteins known to be present on endosomes, and they can say these bits of the other cell that are coming in are in fact taken up by the endocytic pathway.
And an acidic environment encourages that as
Well, that they don't actually look at that. The endocytosis pathway involves acidification.
Yeah, sure.
Whether that's
They-
involved, I don't believe they looked at, but they
They
do.
looked really nicely at the where in the pathway these went, and they showed that it didn't get all the way to the lysosome.
Yeah, so they escape before the lysosome because they're lacking a lysosomal marker.
Okay.
But they're brought in by endocytosis.
Okay.
So these bits of N1 cells, right, go to the N4 cells that get endocytosed, as Rich said, they're vesicles by this point, and then they escape the endosome before they're degraded by the lysosome. And that's how they get into cells. And they look at the nectins that they know from previous work what amino acids on the nectins are important for this interaction, and they can change them. There's a phenylalanine that's very important for this interaction, and if they change it, they can really dramatically reduce the adhesion of the nectins, and it reduces the uptake, the transfer of the cytoplasmic contents as well. So the nectins have to interact by these phases in order to get the transfer. And also the cytosolic regions. These proteins are transmembrane. They have a cytosolic region of a number of amino acids, and those are important for transfer as well. If they take it off, you don't get transfer. And then if they swap them, you can get transfer in a different direction, unidirectional transfer. And so you can get unidirectional. As Rich said, it goes N4 to N1, right?
Yes.
Mm-hmm.
Normally.
Is that right? I get confused.
They've got a nice Punnett square.
Gotta look at this. Right.
They're showing the arrows.
Yes, and
which ones go in
N4
which
to
directions.
Yeah.
N4 to N1.
N4 to
Yeah.
N1.
If the acceptor, if you put the N1 cytoplasmic domain on the acceptor, then you're getting unidirectional transfer. If the donor has either N3 or N4, transfer becomes bidirectional when the interacting cells have nectins that each contain the N1 or lack cytoplasmic domains entirely. So normally it's unidirectional.
Binding to each other, saying, let's go this way. No, let's go this way.
But if you
no,
take the cytoplasmic domains, it goes bidirectional, or if you have N1 on both, it goes bidirectionally.
Crazy.
It's interesting, but
I thought it was a really cool experiment, but at the same time, I also have like 3 favorite experiments in this paper. So
they also
one of many.
They also label the cell proteins with dyes and show that the cytoplasmic proteins are actually transferred when this happens. Mm-hmm. So they're looking at the nectins, but they want to know, are there also other cytoplasmic proteins, so they can label them and see them go over as well. They can look at the kinetics of transfer and so forth. And the protein transfer kinetics are the same as the nectin transfer. So that's pretty cool. All right. So we have transfer from one cell to another mediated by these nectins. Then
could a virus go?
Could a virus do this? That's the cool thing, right? And since they're interested in this because they're working on nectin, they're a measles lab. So they're going to look at measles. So they're going to do a series of experiments where you have a measles infected cell and ask, can you transfer the virus to another cell via this mechanism? And their viruses are modified so they have fluorescent proteins of a different color from the nectins. And so the measles is cyan, which is different from the cherry and the green. Mm-hmm. So you can distinguish all of this, right?
Right.
It's beautiful. It's just beautiful. So now the experiment is they have a cell which can be infected with measles. It's an N4.
N4, right, which measles can use as a receptor.
Right.
And then the N1 producing cells are not infected. And they mix them. And the genome can be transferred into the N1s, and they can see it by the cyan that's produced.
Right.
Yeah.
And they just mix the cells together. They let them settle. And
so the question that everyone would then say, when it's transferred to this new cell with the N1, can it replicate in that cell as well?
Yeah, well, they see the fluorescent protein. Does anyone recall, do they look for infectious virus production in those cells? They don't.
No.
Not that I recall.
So is that a paratenic host then?
What is a paratenic host?
It's a host that's unaltered by the intrusion of a parasite and just it moves it from one place to the next.
I don't think so. I think
these
cells are both the same except for the surface protein. So if the virus can replicate in N4s, it will replicate.
Yeah, but they're going to talk about another cell type soon.
Well, yeah, in a moment. But this just shows you that you can transfer the genome in a functional way from one cell to another, right? That's the key.
Right.
It's functional, which means that it can replicate and produce proteins. But they didn't look at infectious virus. That's true.
Right. To reiterate how they know it's functional, it's because they're getting the cyan expressed, which means that they had transcription going. So it didn't just get there, it got transcribed. So it's intact functionally.
You take an N4, you infect it with measles cyan, then you mix it with N1 cells. The N1s don't have the measles receptor. And then you see the transfer, you see the fluorescence, the cyan fluorescence in the N1 cells. It had to be transferred from, and they do all the controls which show it has to be transferred from the N4 to the N1 cells.
Mm-hmm.
Throughout this paper, they do such a good job of telling you the controls they're doing. And then at one point they even say, this sets up the experiment, that we've done the controls and now this lets us do it.
Yeah. And the N1 cells die.
Yeah.
So another indication that the virus is replicating in them, right? Mm-hmm. They use, so these are these A431 cells. I don't know what they are, but they say we're going to look at some human epithelial cell lines, which are more physiologically relevant And they show the same thing, basically. They can get this transfer in an epithelial line where these nectins are now at physiological levels. They're not overproduced as they are in their experimental cells. The real cool experiment now is, can you do this from an epithelial cell into a neuron? Now, it turns out that mouse superior cervical ganglia produce N4. And so they showed that N4 goes to N1. So they said, can we transfer to, the mouse ganglia have N1, not N4.
There we
yes,
go.
the N1s are the recipients.
The mouse ganglia have N1s. And they did this, they infect N4 cells with measles, and they put it on top of the neurons, but they said the signals were all mixed up and too complicated, so we're going to try this a different way. And they use a chamber system called the Campineau chamber system, where you take these mouse neurons, the superior cervical ganglia, and you put them in a chamber, and you put a ring on top of the cell bodies, and then the axons grow under the ring in the culture dish, and they're essentially isolated from the cell body. So you could do things to the axons independent of the cell body. And Enquist has done this to show transport of herpes viruses. You can put them on the axons, and they get transported to the cell body. So they do this, and the ring sits on top of the cell bodies. The axons grow out. Give them a few days to grow out. And then you can add your cells that have been infected with measles onto the axons, and they show that the cyan measles will get into the cell body.
Amazing. From
the axons.
That's a beautiful experiment.
Yeah.
Very pretty.
And so well illustrated in the figure where they have the
right.
By the way, Alan, is this open access?
No,
it's not. No, I couldn't even get it at home, which I usually can do.
Yeah.
So they have this wonderful cartoon of the ring and the axons and everything, with little boxed insets in red or blue of the cell bodies or the axons, and then the actual photographic data in boxed figures that are red or blue. So you know exactly what you're looking at without reading anything. It's amazing.
Great graphic design.
Right.
Yeah. So you could write to the authors for a copy of the paper.
Right.
Yes.
I'm sure Generous would provide it.
He would. He would generously. You could write and say, could you generously provide? The last experiment is pretty cool. So now remember, we've got these epithelial cells infected. We put them on top of the axons, and the measles gets transferred, right? They say, is this N4 dependent? So in the epithelial cells, do you need N4? How do you answer that question? Because you need N4 to infect. Well, you put the other receptor, the SLAM receptor, into those cells. You can now infect them. You put them on top of the axons. No transfer. You need N4. Mm-hmm. It's cool, right?
Yeah. So these neurons, are these central nervous system neurons?
Superior cervical ganglia. They're peripheral. Right?
I'm just curious as to the natural history of measles. You said 1 out of 1,000 gets encephalitis and dies.
No, they're not.
So it's harder to get these virus particles inside the brain.
It's located opposite the
blood-brain barrier.
So these ganglia are located opposite the second and third cervical vertebrae. So they're not CNS. No. But they're using them as a model.
No, I understand.
Right? Yeah.
Mm-hmm. But I'm just curious how that translates to the natural history of the virus infection.
They talk a little bit about the potential for retrograde transport once the neurons are infected.
The point is it's a neuron, right?
You mean something like rabies spreading through the nervous system?
Yeah, I think the hard part is probably getting into neurons.
Right.
Mm-hmm.
And then from there,
it's all connected.
It's all connected. You could do like a herpes virus, or aren't some of the nectins in the junctions?
Mm-hmm. Yeah, you certainly are. Yeah.
So one of the things that they point out here is that this doesn't obviously provide a mechanism for neuron to neuron transfer.
Right.
So it's not necessarily going to spread.
Yeah.
In the nervous tissue.
So they're thinking if your measles virus goes in your nasopharynx, the virus could get into an epithelial cell that has N4,
Right.
and then they could get into you have neurons in the naso.
Sure.
Of
That goes right back to the brain.
back to the brain. So maybe those epithelial cells could donate
smell receptors.
a bit of their cytosol with measles, RNP to these neurons. Then it would get in. But as Rich said, spread is limited to the first neuron unless infection becomes receptor independent. And they point out that in brains, in human brains, there are mutations that have been identified that lower the activation threshold for viral fusion. And maybe that's why they're selected, so that they can easily fuse from neuron to neuron in the absence of a receptor.
Right.
Okay, so these are isolates from the human brain from people with encephalitis. They have consistent mutations that lower the fusion threshold, which maybe you don't need a receptor for that. Right?
So those are the people that would go on to die from this infection then?
Yeah, people with encephalitis, yeah.
Right. So I think there's a threshold somewhere, because only 1 in 1,000 people gets encephalitis from this.
Sure.
And it may be at this first step, this Nectin-4 step, getting into the neurons, or it might be something, some genetic type that does something in the neurons that makes it easier for the virus to subsequently spread from neuron to neuron. But we're going to need to, there will have to be some more papers to figure out that part of the process.
For sure.
What's really cool here is we didn't know anything about how this might get into neurons. And now here is this elegantly demonstrated mechanism that makes perfect sense.
So people think there's a neuronal receptor, but it's never been identified.
Right.
So maybe you don't need one.
You don't need one. Your neuronal receptor is Nectin-4 on a different cell.
Yeah.
And maybe once it gets into the neurons, then you select for viral genomes that have a lowered fusion threshold in their glycoprotein, and that's enough to get spread.
So we have met the enemy, and they is us.
That can be tested.
Yeah.
The interesting thing is, what about other pathogens?
Yes.
Right?
Yes.
And they mentioned poliovirus, which binds to a nectin-like protein.
Yes.
Which also only causes neurological symptoms in a minority of cases.
Nectin-5.
Racaniello's protein.
No, it's not mine, but they do reference the paper, and it's Nectin-5, and that interacts with N1, so that could deliver poliovirus.
Right.
But I'm more interested in, because we can't work much on polio anymore, and these days are numbered. Enterovirus D68 is a respiratory virus that causes paralysis in a small number of kids, but there's no viremia.
Right.
So maybe this is what's happening.
This could explain a whole bunch of pathogenic processes that occur occasionally that are neurological or not. I mean, it might end up being highly specific, or it might not. This is a mechanistic paper. This is not a this doesn't prove causality, but this is just a really cool door to open.
Yeah, absolutely.
They mentioned
I was sort of
the C. diff tox.
Yeah.
As a possibility.
Yeah, exactly.
C. diff
I was
binds
sort of wondering,
N3. Yeah.
With something, whether they could show whether this requires binding to a nectin or not. You know, they show that when they label the proteins in the cell, that a fair number of labeled proteins are transferred. Are all of those binding to nectin, or is some of this random?
Oh, I think it seems random to me. I think, or I guess that's not proven, but that was the implication to me.
That was my first thought too.
Yeah.
And so wouldn't that mean that pathogens that don't even have anything to do with nectins could be transferred in this way?
Mm-hmm. I would think so.
I like their last paragraph. They say the nect bulk. So this thing is called nect, right?
Right. They dub this process nect.
Tell me what it is again.
Is where'd that abbreviation get broken down?
Elicited cytoplasm transfer.
Transfer.
I thought they could have named it something different, like nicked, induced instead of elicited, and then it would be clear whether you were talking about the process or the protein, but that's just me.
Anyway, they say nect may be ancestral. They say after establishment of the precise and tunable signaling pathways required for cell specialization and tissue function, Nect would have been silenced. But some viruses can still take advantage of it.
A question about the virus.
Cool.
Again, you have measles virus of certain titer, and a certain number of viral particles are attached to the donor cell with Nect4. How many virus particles do you think each cell has to have before you get transfer?
Well, the cell has to just be infected, I think.
It has. I thought it was attached to the Nect.
No, it's not attached. It's getting into the cells.
It's a piece of cytoplasm that goes in,
and then via Nectin-4, and then Nectin-4 transfers it to a Nect1 producing cell.
Right.
Yeah, it's a piece of the cytoplasm.
Okay, that's different.
Exactly.
Different
So
question.
this may indeed be his most important finding, because if it turns out that it is explains a lot of disease pathogenesis, that would be cool.
Yeah.
The word that we've used before in my field that we've used before to describe this sort of passive transfer of something is hitchhiking.
Hitchhiking.
Yeah.
Okay.
Yeah.
So it's as if the cytoplasmic proteins and the virus are hitchhiking along with these vesicles that are being transferred.
Or they're stowaways.
Next one to the next one. Stowaways. That's good.
Because you use the word cargo, I would just think about stowaway then.
Sort of like it's Trojan virus.
There you go.
There you go.
Mm-hmm.
So he wrote to me. He had come here a couple of years ago and given a talk on this preliminary data. You remember some data from a seminar I gave at Columbia about 2 years ago. We now understand how and when cytoplasm transfer happens. We can reverse the direction of flow. The intercellular streams of cytoplasm are transient but can carry along measles virus. Nect can transfer measles virus to neurons and could possibly be the first event in the development of SSPE.
That's subacute sclerosing panencephalitis,
Yes.
by the way.
Long-term
people out
sequelae of measles.
It doesn't think I know anything about anything.
Who said that? Who said
You
that?
did.
Never. Listen to his last sentence.
No, we think you don't know anything about everything.
Oh, thank you.
No, I got that wrong. Wait.
No, you got it right, Al.
Roberto writes, I wonder whether Nect could be material for discussion in TWiV. Measles virus spread hits a nerve. It's his suggested title.
Well, how could we dare disagree with the author?
Roberto. Yeah, so he talked at ASV, of course. He did a paramyxovirus satellite, but what did he call it? A satellite outside of our solar system because it wasn't an ASV satellite.
Satellite presentation.
So we have satellites at ASV, which are like before the actual meeting. It's the day before. And so he said this is a extra-orbital satellite.
Prodromal. Prodromal. You have prodromal.
So if I could just toss out here.
Yeah, sure.
People think that satellite is not a good name for those sessions.
Really?
So we might be looking for some other name. And if they are interested in helping us brand them differently, we're open to suggestions. So just send it to me or council. Yeah.
Why not prodromal?
Pre-show, prodromal.
The prodromal presentations. Yeah.
How about a viroid?
Because eventually they will all become viral.
Yeah, well, send in your suggestions.
I just did.
Yeah.
What did you suggest?
Prodromal.
Prodromal?
Yeah.
We're talking about meeting sessions. We want to, make them modern and
not a
something
disease.
that everybody will be interested in going to.
People can get very creative. That'll be fun. All right. Let's do just a couple of emails here. We have one from Colin. Dear TWiV hosts, I have recently become addicted to your podcast. I've had an interest in infectious diseases for a long time, but without much of a background in microbiology or virology. I've learned so much from your weekly discussions, which have helped me tremendously as I delve into my PhD studying dengue viruses. I want to make two remarks. One, as you covered in TWiV 535, miles to go before I leak, Flavy NS1 is a hot topic. I think we're only beginning to scratch the glycocalyx surface of how NS1 interacts with the host. Some comments. 1. Glycocalyx breakdown products are a pretty crude measure of vascular leakage. Many different ways of causing vascular leakage will shed these glycocalyx components. How do you determine how much of glycocalyx breakdown is due to NS1 versus cytokines? This is especially difficult because the breakdown is transient, as shown in their TIR assay, transendothelial and electrical resistance.
Close.
Is it epithelial or endothelial?
Depends what kind of cells you're talking about.
Yeah.
Okay.
It can be either.
So good. I'm not necessarily wrong. The glycocalyx reestablishes in a matter of hours. Good point. 2, the paper did not discuss lipid composition of NS1. Parents, this is not criticism. Our lab thinks that this probably has a role to play in pathogenesis as well, as we briefly suggest in a paper we just put out. I would be happy for you to critique this paper. It would certainly continue an arc. Paper is in Science Translational Medicine. It is a mutation in the dengue virus NS1 protein, which I would critique as being incorrect. Okay, and is associated with greater disease severity in mice.
The mutation is in the gene.
Yes, I would say a T164S amino acid change in the dengue virus protein.
Okay.
That would be good, right?
Yes, right.
So this is interesting. So in people with severe dengue, they consistently find this amino acid change in NS1, and so that's documented very nicely in this paper in cells and in mice, for example. And they suggest that it has something to do with lipids, lipid composition of replication complexes. It's pretty interesting. All right. On a different note, I recently attended a Keystone conference on positive sense RNA viruses. While I learned a lot, one moment stood out to me as the conference made efforts to bring people from underrepresented backgrounds and from countries where many of the discussed viruses are endemic. I was heartened to see a significant number of African researchers, although none of them were selected as speakers. After Priscilla Yang had delivered a talk on antiviral strategy, a lady professor from Nigeria, I'm guessing here, I don't know, stood up to make a call to arms for researchers to focus more on antivirals and diagnostics, especially for the aforementioned countries. Awkwardly, there was no further acknowledgement of her plea as the moderator simply went on to the next speaker. Boo, moderator. I'd love for the TWiV team to comment on this issue. How well do you think our scientific community is balancing between the real-world need for therapies and the need to publish? I understand we need both basic and translational medicine to achieve this goal. For that matter, does the TWiV team feel that the overall quality of publications has improved over time? But I can see how people in need of therapies may despair at how much money is being spent on working out the molecular biology of some obscure protein without any demonstrated impact on pathogenesis, for example. Anyway, thanks once again for an awesome podcast and the drive to communicate science. I do hope that this letter will remind us of the big picture of many of the viruses that we are studying. And Colin is a PhD student at the Duke National University of Singapore Medical School, where in Singapore it's over 30 C all year round with our monsoon season from December to March.
She's not kidding.
Dickson, monsoon season? You want to send me there in the monsoon season?
I have nothing to do with this. I was there twice, and I wasn't there during the monsoon season. So
okay. So Kathy, would you like to say something?
Oh, just about the moderator moving on and so forth, and that I think it is difficult to balance the basic and translational needs. And I wish there were a way to have more kinds of input from people in those other countries. And to that end, ASV is working on expanding our Latin American Scholars Program to be more global. So watch for that. But it would have been awkward to be there. I agree.
I think a moderator needs to say something. That's part of being a moderator, right? Not just move on. That's too bad. But the many people are establishing capabilities in other countries so that people can do research there themselves. Instead of going and taking reagents and bringing them home, they establish capability. That was done in Africa during the Ebola outbreak in West Africa. That's been done in South America. And I think that's really important to empower people to be able to do research. But here in this country, it's really hard to do antiviral and vaccine research, because if you're R01 or a grant-driven laboratory, it's really hard to get grants to do that sort of thing.
Yeah.
Because you could say, we're going to look for antivirals, and they'll say, well, how do you know you're going to get them? And that's the end, right? So it's tough unless you, so Priscilla Yang, I know, is part of a program, an NIH program, which is focused on getting R01 investigators to look for antivirals. Okay. I was actually on the advisory board of that program, and I forgot the name of it, is they encourage people to get antivirals. And that's really important. I think we need more of that, because otherwise it's, we can design experiments to look at a single protein, as Colin says, and that's much, much easier to get support for than to do antivirals and vaccines. Anyway.
I agree. I think that sometimes people should also remember that when we learn about those obscure proteins, we might find out about a new antiviral we never would have thought of before.
Yeah, you never know, right? Yeah.
And so we've got to balance both sides of this.
I think the government makes another assumption, and that is that industry will take care of the antivirals because there's money to be made in it. And so you just stick to the basic stuff. And that's what the RR1s, and the RR1s are generated by groups of scientists that have RR1s. So it's self-fulfilling, basically.
Of course.
I'm going to take a controversial position here. I don't think we need more basic researchers to shift their focus to applied research such as antivirals and diagnostics. And just to put some perspective on the scale of the investments here. So Colin is expressing a very widely held view that a lot of money is being spent on basic research, and maybe that money could be redirected to more practical ends. And that's a reasonable thing to think, but when you look at the numbers, the current NIH budget is $39.2 billion. The current Pfizer market capitalization, this is a single pharmaceutical company, a large one, but a single pharmaceutical company, is $239.5 billion.
Wow.
So the amount of money being spent on coming out with the next Viagra,
that's why. I mean,
is dwarfing the amount of money being spent on the so-called search for the molecular biology of the obscure protein. But without that basic research, you're going to stall out on the applied research 20 years from now.
You're not going to find the next target.
You're not going to find the next thing for the next disease. So you need to keep the basic research going. And there are problems with the basic research funding models, many of which we've talked about here on TWiV, and I'm in agreement with a lot of the critiques of that. But I don't think that a lack of focus on applied research is one of those problems. In fact, if anything, I think there's been too much of a push in recent years to emphasize translational research for scientists who had been doing basic research. Because what we found is, and I mean we as a society, is we found that there's this valley of death between finding a promising lead and putting a pill in a bottle. And you get to a point where we've got lots and lots of promising leads for therapies for maybe not so many antivirals, but cancer therapies where we have promising leads. It's ridiculous. There's a huge pile of them. And then pharma companies will take some of those and try to pursue them, but they're picking blindly. And the pharma gets a lot of criticism, some of it quite justified, about their pricing policies and that kind of thing. But they do pump a lot of money into trying to develop drugs. And they invest in this really expensive step of getting something from working in mice to actually being a drug that you can put into people. And that's a really hard problem. And that's where we're stuck, is that gap between what we found in the research labs and what we can actually turn into drugs. And so I fully understand and empathize with the professor from a country who's being struck by these horrible infectious disease problems, wanting more focus on developing more strategies to deal with them. And yeah, we do need that. But I don't think that the basic research community is the right group to come up with those solutions.
Yeah, the axiom, I guess, is the further away you live from the real problem of the disease itself, the more likely it is that you'll be interested in some what other people would consider esoteric portion of the biology of certain diseases. And that's
if you have the luxury of doing that, right?
Yeah, that's where the frustration comes, because
it may end up being that the answer to this comes from the people who are affected. I mean, there are nations in Africa that are, I think, making, or at least trying to make, better investments in innovation, and they may be the best place to try to pursue this, and that's the kind of thing we should encourage.
Our meeting, the American Society for Tropical Medicine and Hygiene, has a whole section on viruses, and when the people come to talk at that meeting, they are coming from the epicenters of where these virus infections are raging, and that really galvanizes the audience to realize that those are unsolved problems, even though you're working intensely on, the receptor molecule or something like that.
All right. Anything else? Many drugs are
not from me.
Many drugs start in some way in academic labs.
They do,
of course.
NIH funded.
Absolutely.
I mean, even not the compound itself, but this, the target or some concept. Mm-hmm. And so I think we need to keep that up. I agree.
Sure. Whole companies are founded on those concepts.
Sure.
Right. That's right. Alan, can you take the next one?
Sure. Jason writes, Dear TWiV crew, at the end of TWiV 554, you related a story about how Bernard Seltz promoted the idea that people should be able to choose whether to vaccinate or not. As part of that discussion, you posited that the Seltzes don't care about other people's children. While I would agree that his statement is wrong and dangerous, I don't agree that this means he doesn't care about children. On the contrary, I think in many cases an anti-vaxxer is passionate about their cause because they care about the kids. I will admit I don't know if Seltz is anti-vax, but the commentary would seem to lean in that direction. You followed up that discussion with an analogy to traffic laws and car seats. I think this is spot on, and I wished you took it further and mentioned that breaking these laws has consequences in the form of monetary and legal penalties. Should fines be considered for those that refuse to vaccinate? As always, I enjoy the show, and while I can't follow everything, I do end up learning a lot. Thanks. And, Jason signs as Jason Xenophage.
And the concept of the innocent bystander comes to mind when all of that is discussed, because if you don't vaccinate your own kid and they come down with something and they give it to all the other kids at the daycare center, that's not a good deal. Or to the adults that haven't been vaccines were off.
And wasn't there a one of the provinces in Australia, or maybe it was nationwide in Australia, they implemented a policy where people who didn't vaccinate their kids were going to get penalized financially?
Yes.
I remember we discussed this on TWiV, and I don't remember all the details, but there was definitely a policy put through to that effect, and I support that.
So it says breaking these laws has consequences, not just in the form of monetary and legal penalties. You could die.
There's that too.
You could go right through the windshield of a car.
What would actually cause people to act, because explaining the risks has not done so.
I know. I remember seeing a Charles Addams cartoon when the seatbelt laws came out, and it shows a couple sitting in their car, desperately trying to get their seatbelts unbuckled as a train is bearing down on them. They're stuck on the rails. So they can't get their car started. So that would be the one exception. I would say not having a seatbelt would have saved your life then, but that's what Charles Addams.
But then you have to ask, how the heck did you get stuck on the train track?
That's true too. That's true too.
Yeah. So his point about Seltz is right. I don't know if maybe they do like kids and that's why they're doing that. It could be. And maybe parents who are worried about vaccines care for their kids and that's why they do it. I just think they're misguided, right? And so the Seltzes are misguided as well.
Mm-hmm.
You missed the opportunity, Alan, of reading the following.
Oh, so there's a quote from The Hitchhiker's Guide to the Galaxy at the end of the letter. There is a theory which states that if ever anyone discovers exactly what the universe is for and why it is here, it will instantly disappear and be replaced by something even more bizarre and inexplicable.
Right.
There is another theory which states that this has already happened.
Very good. Very good. Penalties for not getting vaccinated is interesting. I can't imagine that this country would ever pass such a thing.
Not at the federal level, but I can see it happening in some states.
In some states, maybe, yeah. California, for example.
Some states don't have helmet laws.
Right.
That's insane.
Yeah, that's crazy. All right, let's do some picks.
Hey.
Brianne, what do you have for us?
So my pick is from the ASV show. This is what I had planned on picking then. At ASV, we were honoring Bill Yachlik, who had been involved in the founding of ASV and who recently passed away. So my pick is a YouTube video that came from his department at Duke that talks to him and about his career and his legacy. And so I think it's really useful and really interesting to hear about Bill Yachlik and what he's done, both from him and from his colleagues.
This is nice.
It's really nice.
It's a nice video. It's very well done.
Cool. Yeah, this is from last week.
Yes, it is my pick
Very
from last
cool.
week.
It's cool. Thank you, Rich. I was just trying to find out if you had left us. I haven't heard from you, but you're.
No, I'm here.
Very good.
I'm listening in rapt fascination.
Not in the mummification sense of the word, I hope.
Right.
Alan, what do you have for us?
I have a photography award thing, and I know Dickson and I occasionally pick these, but this is from the Audubon Society, so it's birds.
Nice.
And these are the 2019 Audubon Photography Awards, and I think they are spectacular.
Yeah.
Just amazing images of wildlife and things that I didn't even know occurred, like this grand prize winner with the red-winged blackbirds singing on a cold morning, and it's making
That's great.
It's making condensation rings because it's puffing air out as it sings, and she caught the rings, and it's just this amazing image.
This one with the eagle, my wife showed me
Yes.
this morning. She said, "Did you see this eagle? So a fox had caught a rabbit, and then
It was about to,
and the eagle grabbed the rabbit, and the fox, I guess, grabbed the rabbit or wouldn't let go. So they're both up in the air.
The eagle is actually holding. If you look closely, the eagle was holding the fox and the rabbit,
And the rabbit.
Couldn't make
and
up
then
its
dropped
mind.
the fox from 20 feet. And the fox was apparently fine, but hungry. And the eagle flew off with just the
rabbit. Poor rabbit. To which I said to my wife, "Oh, poor rabbit." And she said, "Nature's that way, you know." Food chain, right?
That's right. That's right.
The eagle picture reminds me that when these raptors get fish, they orient the fish to be aerodynamic as they're flying away with them. That blows my mind.
Have you ever seen, though, Rich, a picture of a osprey that hooked too big a fish to lift out of the water?
No. What do they do?
I think I have seen this.
They have circle hooks for claws.
Yeah.
Almost. They almost come back and touch themselves.
Mm-hmm.
Sometimes they drown because the fish takes them under the water.
They can't let go until they eat it, right?
They can't let go until they get out of the water. That's right. That's right. Go ahead.
I was going to say what I like about these awards is that they have the story behind the shot where the photographer tells something about the photo.
You're going to just love my pick then. No, really. I'm not next, or am I next?
You could be if you want to be.
No, well, it's a good follow-up.
Okay, go ahead. What do you have for us, Dickson?
I have a small snippet. You'd call it a snippet of a documentary. It's a small video from National Geographic of the discovery of the true pollinator of the ghost orchid. And if you think for 1 minute that, it's Sunday, why should I go back to the lab? No. Go back to the lab. You have to do it. It's Christmas Eve, for God's sakes. I've got my family with me, but I have to feed the cultures. I have to go back to the lab. And everybody has a family like that. They grow up understanding all of that.
Maybe
a nature, your second family then. The nature photographers of the world, the ones that are really dedicated to this as a passionate addiction almost, you'll see what perseverance can do for you. This is a fabulous, sort of an Aesop fable of perseverance wins the day eventually. And, of course, technology as well.
These are cool.
And it's National Geographic. So
and it's National Geographic, of course. I mean, they would reject all the ones that weren't.
Right.
But it's amazing how long it took to actually find out what does pollinate that flower. It's got to have a tongue longer than the actual organism, and indeed it did.
Interesting.
Yep. Yeah. Rich, what do you have?
So I have more follow-up on the honoring Bill Yachlik theme. I was we had a mission and limited time, I would have liked it if we could have said a little more about Bill's science. We talked mostly about the establishment of the ASV, and the YouTube video that Brianne picked goes a long way in doing that. I have just a couple of more focused things, two little picks. So Bill's research, a lot of it was on pox viruses, which overlapped with mine, and so I knew him. He used to come to poxvirus workshops, and some of his early molecular virology studies were really seminal in the field. And I've just picked, if you look through a list of his publications on PubMed, he touched on molecular points of the whole virus life cycle in poxviruses early on. And I've just picked one, this JMB paper that I referred to all the time, called Hybridization and Sedimentation Studies on Early and Late Vaccinia Messenger RNA, which if you ever had the inclination to look at a historical basic paper, this is a great one. It uses all techniques from the 60s and is absolutely prescient in understanding the transcription in poxvirus infected cells. It's just really a great classic study.
And is absolutely not open access.
And is absolutely not.
Sadly.
Still?
Sorry.
Sadly.
Wow.
Yeah.
It's Elsevier. It's, yeah.
Too bad. So the other is, people who do a fair amount of tissue culture, in particular with suspension cells, may be familiar with using the Jochlik modification of minimal essential medium.
Yep.
Formulated by Eagle. And I just thought I would point out that we're talking about the same Bill Jochlik there. So he goes right into the fundamentals. He spent time in Eagle's lab. I forget whether it was as a postdoc or whatever. And I presume that, I don't know that this medium necessarily came out of his time with Eagle, but certainly he was familiar with all of Eagle's culture studies, and ultimately a lot of poxvirus work was done on suspension cultures. So he did some modification of suspension culture media to grow HeLa cells that became embedded in history as his modification of the culture media. To this day, when we purify vaccinia virus, we reference a 1960 some odd paper of Bill Jochlik's for the purification. So
cool.
Kudos to Bill for his science.
Yes.
Mm-hmm.
So,
Rich,
you have a particular fondness for eagles?
I guess so.
You picked out two of them in this picks of the week. Quite different types of eagles.
Beetles.
Eagles.
No, but the original was beetles, right?
That's
Right.
God must have had it.
Okay. Kathy, what do you have?
Well, mine is also from last week, and it's semi-related to the fact that we like a lot of things about scientific art. And at ASV 2018 in Maryland, in part because we were in the DC area, we were able to attract 4 sci art exhibitors to the ASV. We didn't have any of those this year in Minneapolis, so I picked a sci art thing that's crocheted Petri plates, and these are absolutely
great.
Amazing. She puts them in glass plates, and the media itself is felted wool, and then crochets these amazing molds and things. And so they're available online at several sources. They're not inexpensive, but I suspect a fair amount of labor goes into making these.
Yeah.
So, they're really cool. There's fuzzy ones and all different colors and textures. And
They're amazing.
Yeah.
They're beautiful.
I just really like them.
They're very cool. Yeah, they're expensive, but beautiful.
Nature's abstracts.
Really nice. So it's 9-centimeter Petri, 220 pounds.
Mm-hmm.
But gorgeous.
Yeah.
They are
Nice.
remarkable.
That's
Cool.
the British price, not the shipping weight.
Right.
Right. Exactly.
Yeah. My pick, is also from last week's episode at ASV. It is a document which is on the ASV website. It's called Documents Relating to the History of ASV, which many people may not find because it's kind of buried. But it's pretty cool. It's called The Founding of the American Society for Virology, honoring Bill Yachlik for his vision. They have a copy of the paper, How the ASV Was Founded, which we talked about during the ASV episode, which is pretty neat. It's got the multi-step growth curve showing the increase in membership of ASV.
Right.
It's got some of the initial founding people. But what I thought was cool, and there's other stuff as well, letters and correspondence and so forth from Bill Yachlik and other people. If you scroll all the way to the end of it, I believe, yes, there's a reproduced article from ASM News, November 10th, 1981. It's from the president of ASM, who at the time was Fred Nehart. It's called ASM Scene. A special message from the president. An issue of some consequence to ASM has arisen. The plan of some of our members to form a new organization of virologists. Gosh, he makes it sound so evil.
Viruses are evil.
Not all of them.
I thought you said this was a show about the kind that make you sick.
It was 10 years ago.
What was the exact founding date of ASV?
June 9th, 1981.
1981.
Were you born?
You know better than to ask a question like that, Vincent. You weren't even haploid.
The reason this is cool is because
I think we were all born then.
we were all born.
Yes.
ASM, all virologists were members of ASM. So ASM was worried that they would all leave, which they didn't. They went to their own meeting, of course. That was the whole point. And so they were worried because ASM is always, it's a great society. They do good things, but they're always about money because they're big and they have to pay for things. So this reflected that concern.
Including some episodes of TWiV, and we thank them for that.
We do. And as Sid Grossberg said, this came up during the TWiV. Please remember, they support ASV every year. Mm-hmm. Right? And Kathy, they make contributions.
They're hoping you come back.
Yes, well, we're not going back, but many of us are still ASM members,
right? And we're continuously trying to strengthen interactions between the societies.
Right.
Exactly.
All right, I have 2 listener picks, but I'm going to skip one again because I want to spend more time than we have on it, and we will just give you one from Neva from Buda, who sent an article from Ars Technica. In the not-so-distant future, synbio could lead to global catastrophe, maybe. Come listen to a TED Talk and then discuss the darker side of synthetic biology.
I'm going to assume, she didn't write much with this, I'm going to assume that she sent this thinking, get a load of this bozo, okay? Because this is a journalist who is going off the deep end of catastrophe scenarios involving synbio.
Yes.
Yeah.
And he starts out by, there's a paragraph here. In 2011, 2 separate research teams, one in Holland, the other in Wisconsin, set out to repair a defect in what he calls a defect in H5N1. By carefully manipulating the bug's genome, they soon had something just as lethal as the classic edition, but also wildly contagious.
Wrong.
Wildly wrong.
It's not as lethal. It's wrong.
It goes on and on. So this guy doesn't have a clue what he's talking about. Not a clue. But he's out there on TED basically publicizing himself.
Yeah.
Well,
that's what TED Talks are.
Yeah.
And this is typical. It's
I resemble that remark.
This is 2019, and this story is brought up and up over and over, rehashing the same wrong things about it. And people who are not scientists read this and they get scared about what we're doing. And they don't read what we say because we don't give TED Talks.
Right. Well, not all of us, but some of us did.
Yeah, but you did it about plants.
This is Well, I did it about something else, not me.
You couldn't get to a TED Talk where you said, this is okay and don't believe this guy. They don't care about that, right?
Right.
But yes, you're right, Rich. She didn't say anything, but she meant this is ridiculous, and it is. It's crap. I don't know why you had a TED Talk on this.
No, I'm sort of snickering because it talks about superbugs a lot. There was a recent book published called Superbugs, and I wrote it in a list of things I wanted to look up in my phone, but I typed it wrong and wrote superhugs. And so now every time I see superbugs, I think about superhugs. That must be much nicer.
I thought it was a customized VW bugs.
The benefits of all this research far outweigh any possible negatives. I mean, that's why we're around and we live so long, right? Why do people have to trash it? All right, TWiV 559. You can listen on any podcast player. Please subscribe. We would like you to do that. Push the subscribe button so you get every episode, and we find out how many people are listening. Send your questions and comments to twiv@microbe.tv. And if you really like us and our other science podcasts, please support us. You can give $1 a month over at microbe.tv/contribute. Why do we want $1 a month? Here's why. We have monthly expenses. We would like to do more shows where we all get together in different places, like ASV. 3 of us were there, right? 1, 2, 3, 4.
4.
I always get the count wrong. 4 of us were there, but there are other TWiVs we'd love to do and other podcasts. Bring a couple people along. We'd need your money to do that. So think about it. Microbe.tv/contribute. Dickson Despommier, thelivingriver.org, Racaniello.org. Thank you, Dickson.
You're welcome.
Thanks for coming in.
It was great. Yeah, it was good.
This is good.
It was good.
Brianne Barker's over at Drew University, BioProf Barker on Twitter. Thank you, Brianne.
Thanks. It was great to be here.
That mic sounds so good.
It does.
Mm-hmm.
It's like 3,000% improvement.
Like you're in the room with me. Right
It's so clear. It is so clear. It is really good.
Well, I'm glad. You all sound really good, too, with these headphones.
Good.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. I just want to say one thing. In the pick that you made, the letter from Fred Neidhart, who was a department chair here in my department, is actually closer to about a fifth of the way down the whole document if you use that scroll tool in a PDF viewer. So not at the very end. It took me a while to find it. Yeah.
It's after all the letters from Click and so forth. Yeah. Thank you, Kathy.
Yeah, thanks. It was a lot of fun.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at turbidplaque.com. On Twitter, he's Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank ASV and ASM for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.